And on that note, I'm going to invite the next group up to the stage. So Catherine Howe is going to talk about her work in epilepsy, and Sarah Stevenson and Scylla Jenk are also going to join her. So over to Catherine.
Thank you, Vicky. So epilepsy is an important childhood health problem for a number of reasons. Firstly, it's common. 1 in 200 children has epilepsy, and that equates to 70,000 children in Victoria alone. Secondly, it's very heterogeneous. There are many different types of epilepsy, and these vary in severity. Some are mild, with occasional seizures that children grow out of. But many are much more severe and are associated with extremely chaotic brainwaves that disrupt all aspects of brain function. These children spend a lot of time in hospital and are at risk of dying from their epilepsy. There are many different causes. In the last 10 years, we've come a long way in understanding the causes of epilepsy, but still over half of children remain without a diagnosis. The causes we do know about include genetic disorders that we can identify with genomic testing, and disorders that cause abnormal structure of all or part of our brain, which we can diagnose with MRI scans, although they're often very difficult to find. Structural abnormalities affecting part of the brain are sometimes referred to as a lesion, and the most common of these you'll hear more about later. There's a brain malformation called focal cortical dysplasia, or FCD. We have many different treatments, including anti-seizure medication and dietary therapies, and we also use surgery as a treatment for lesional epilepsies. But unfortunately, there's a really big difference between what our treatments can do and what we need them to do. 30% of children have ongoing seizures despite treatment. So that's 21,000 children in Victoria alone living with the life-changing impact of uncontrolled seizures. There are many people at MCRI working together on epilepsy, and our teams include clinicians, imaging scientists, and laboratory scientists. And today we're going to present 3 examples of how our collaborative work is improving lives. The first 2 will cover how we're working to better identify the causes of epilepsy. We want to make more diagnoses, make them faster, and we want to use this knowledge to guide treatment. And the last example will show how we can use insights from our patients to guide the development of new treatments. So I'll begin with our work on improving the use of genomic testing in infants with epilepsy. So infancy is the highest risk age for developing seizures, and infant epilepsies are the most severe of all of the epilepsies. Seizures are often treatment resistant. Many infants have major developmental problems, and these worsen the longer seizures remain uncontrolled. A one-size-fits-all approach to treatment is not effective, as the optimal treatment typically depends on the underlying cause. We want to be able to give each child the best treatment for their condition early, but with the current clinical standard of care, we're not able to do this. Most infant epilepsies have a genetic basis. There are over 800 causes now known, and genomic testing is really effective at finding them. But in clinical practice, genomic testing typically takes many months, which delays opportunities to individualise management and improve outcomes. In the last few years, the Australian Genomics Acute Care Flagship has shown us the enormous impact of rapid genomic sequencing in critically ill children in the intensive care unit. And our recent work has taken rapid genome sequencing outside of the ICU, and we've used it to diagnose the cause in infants with new onset epilepsy. Our study, known as GeneSteps, was conducted at 4 paediatric centres across 3 continents and had some really important findings. So firstly, we showed that rapid genome sequencing was feasible outside of an ICU setting, and we reduced the time frame from testing from many months down to 3 weeks. Secondly, we showed that genome sequencing made many more diagnoses than gene panels and exome sequencing, which is the technologies we typically have access to in clinical practice. But most importantly, making a rapid genetic diagnosis had a huge impact. A positive result changed clinical management in almost all children. And for over half of children, this meant we were able to individualise their treatment. So the use of rapid genome sequencing has completely transformed our clinical practice at Melbourne Children's Campus, and our patients are benefiting from this in real time. So I want to give you 2 examples of what this means. This is Levi on the left. His seizures began at day 3 of life. They occurred many times a day and were ongoing despite medication. He couldn't breathe or feed independently. But making a genetic diagnosis meant he was able to be put on the right seizure medication, and this stopped his seizures. He got out of hospital, stayed out of hospital, and moved forward in his development without incurring months of damage from seizures. And children like Levi are typically never able to sit, but he was able to walk just after his 2nd birthday. For Stella, the genetic results were a critical piece of information in determining that she would likely benefit from epilepsy surgery. But why is this so important to know early? Well, if we're able to operate during infancy, the developmental outcome is vastly better. A child can regain some of what they've lost, up to 30 IQ points, as you can see on the graph on the right. But if we operate later, we prevent further decline, but we don't gain back what was lost. So moving forward, we want to move rapid genome sequencing into clinical practice around Australia and the world. We want to see it being used for other time-critical conditions that are predominantly managed outside the ICU. And finally, we're working to understand the causes in infants who remain undiagnosed using omics approaches you'll hear about this afternoon and advanced imaging techniques like you'll hear about next. So with that, I'll hand over to Dr. Sila Gench to talk about her brain imaging work and how it's transforming lives. Thank you.
Thank you, Kat, for the great introduction. So as Kat mentioned earlier, 30% of children with epilepsy have ongoing seizures despite medication. So treatment can be surgical to remove the abnormal brain tissue that's causing these seizures, but this relies on really good brain imaging to enable precise diagnosis and also enable planning safe surgery. So I'm going to walk you through a case. This is a typical MRI scan. This is a 9-year-old girl's scan. She has drug-resistant epilepsy, and that means she has many seizures per day that cannot be controlled with medication. Somewhere in her brain is an abnormal region causing these seizures, and using conventional imaging like what you're seeing here can make it difficult to identify. And even if it's found, like this abnormal brain region, even if that's found, surgery is the ideal treatment, but it may be too risky. I'm going to show you how we do things differently at RCH and MCRI. So our research integrated approach allows us to get better imaging. Since 2017, we've been acquiring FDG PET data, which helps us look at glucose metabolism in the brain. And this is typically reduced in seizure-causing brain regions. Since 2020, we established our NACIS group, which is a translational unit in the Department of Neurosurgery, dedicated to advanced image processing for these clinical cases. And we've developed a multitude of tools that allow us to anatomically map the brain. And in this example here, we're looking at the asymmetry of that glucose metabolism. And in white, you see the region with maximal asymmetry, making us believe, giving us information that that seizure-causing region is there in white. We then perform language fMRI, so children do a thinking task in the MRI scanner, and that allows us to obtain information on the localization and lateralization of language function in the brain. And then we generate these structural pathways to determine areas that are at risk during surgery. So in green, you've got arcuate fasciculus, which is important for language processing. And in blue, we've got the corticospinal tract, critical for motor function, so movement of arms and legs. And you can see that lesion that we've identified is nestled in that really critical area of language and motor function. So we need a very detailed surgical plan guided by the neurosurgeons. So finally, once we've got a target area, we generate these 3D visualizations of the brain surface and vessels to enable neurosurgeons to develop a safe surgical roadmap in order to avoid very important blood vessels and those areas of language function and motor structure to enable a safe surgery. So I've just put a little red box around that lesion that was identified, or the abnormal seizure-causing brain region that was identified. And now we move on to what's seen in surgery. So this is the interface that the surgeons see. We've developed a tool that directly loads these maps that we've made into surgery, and it helps the surgeons navigate in the brain like a GPS to try and avoid these major structures and help avoid doing damage to these important areas of language and motor function. In addition, clinicians obtain detailed electrocortical recordings, and they monitor electrical activity in that region identified as being seizure-causing to confirm that's where the seizures are coming from and remove it safely. So just to go back on that image that we saw earlier, I'm going to quickly move on to what her brain looks like after surgery. This tiny little bit of grey matter was removed, and that led to her going off her medication, having no more seizures, and she's currently 12 years old and doing very well with no impact on her language function and motor function. So this really, really highlights our impact in improving our detection of these brain regions that are responsible for this epilepsy, and enabling our surgeons to have access to or plan safer surgeries. So I'd just like to hand over to Dr. Sarah Stevenson.
Thanks, Scylla. I'm going to briefly talk about the way we're using genomics and molecular biology to better understand the brain biopsies that are taken during epilepsy surgery. So here you can see a photograph of the surface of a child's brain at the very beginning of surgery. The outer circle represents the entire abnormal lesion, and the inner dotted circle represents the most abnormal part of the lesion that Scylla just identified with her techniques. In most epilepsy surgery centres around the world, the neurosurgeon would remove the entire lesion and potentially also some normal brain tissue. With these lesions nestled in parts of our brain that are controlling movement and speech, that can lead to very severe consequences for these young children. The team at RCH and Murdoch Children's have been using electrical recordings during surgery and demonstrated that actually the seizure begins in the most abnormal region of the tissue, and then it propagates out to the rest of the brain. And therefore, you can actually remove only this most abnormal part of the lesion, and the child is seizure-free. We've been working with them to look at what the underlying histopathology is of these lesions, and we've identified that there's a key difference between the region that initiates a seizure compared to the region that propagates that seizure, and that's a high density of these abnormal hyperexcitable neurons. And this has led us to hypothesize that it's a cluster of abnormal neurons that's required to initiate seizures. And consequently, we've started to now focus our research on looking for genomic changes in similar types of lesions. So what you can see here is some single molecule detection, indicating that mutations are identified only in the brain tissue of these individuals. This work has demonstrated that there's only a handful of genes that are actually causing this type of pathology. And when we look even closer at it, we notice that there's a varying amount of the level of gene changes in the tissue. And if we align that then with the histopathology findings and Scylla's MRI imaging, we can see that the gene change overlays with the imaging and overlays with the histopathology, providing very strong evidence that these are the pathological substrates that are initiating and causing seizures in the brains of these individuals. So now that we understood that all of these genes that were identified functioned in the same molecular pathway, we had an opportunity to apply some new genomic techniques to understand them further. This is an image that represents over 100,000 human brain cells from mid-gestation until adulthood that are assayed using a single-cell genomic technology that tells us how cells are functioning. As you look around this room, you can see that many of us are signalling our ancestry, personal and professional identities through the way we present to others. We are all interpreting this information to understand both the individuals in the room and the group as a collective whole. Well, cells operate in a very similar fashion. Each type of cell within a tissue presents its genomic information uniquely. And we can look at this information to understand how the cell functions as an individual, but also how it functions collectively with other cells. This is an image of over 100,000 human brain cells from children who have had their seizures controlled by epilepsy surgery at the Children's Hospital. We've conducted a thorough analysis of the differences between this tissue from the children with epilepsy compared to normal brains, and identified a region of pathways where we think that we can intervene. So if I was on campus right now, I would be expecting a phone call from the epilepsy neurologist to tell me to head to theatre, where I would run down with a little bottle of artificial CSF, and I would wait and watch as the epileptologist and the neurosurgeon use the techniques that Scylla showed and electrophysiology recordings to plan the surgical approach. And once they'd finished planning that surgical approach, they'd hand me the very first bit of tissue to come out of that child's brain, and I would then take it very, very quickly to the lab. And that's because we've used this single cell technology to start to identify drugs that we think can actually intervene and change the phenotype of these disease cells to make them look more like normal neurons. And this is ongoing work that we are finishing up at the moment. So it's our hope that we can use these kind of approaches to treat the children that don't have access to the surgeons that our children have, but also treat them while they're waiting for surgery. So I'll move on more broadly to bring everything together. I hope today that we've left you with an impression of how the Melbourne Children's Campus we're embedding research questions into the clinical practice. In the future, we look forward to a whole-of-system integration of genomics and advanced imaging into clinical care, unification of clinic and research to diagnose, understand and intervene for these children faster and better, and that precision medicine will be an option for all children who need treatment. Thank you.
Thanks, team. Just to remind you, if anyone has questions, to come up to the mics and state your question. But before we get questions, I just would like to make a historical comment. So I worked in the epilepsy team in the 1980s and 90s when we didn't have MRI scans, let alone all of the complex equipment that you have. And kids did really badly in those days. And we weren't even thinking about how we would understand what was going on. And I think there was some great innovation in the then team to do things like create a biobank, fund a new MRI scan, and it's really led to this team and their work being absolutely stars in their field internationally, and they're very humble, but many families come from many, many thousands of kilometres away to have their child assessed and treated with the RCH and MCRI work. So any questions? Okay, I have a question, and my question is for Sarah. And Sarah, I've written it down because some of this is above my pay grade. So can you tell me about the benefits that you see in providing a genomic diagnosis over and above what's been done traditionally?
So traditionally for epilepsy surgery cases, individuals have histopathology performed, which is H&E staining, so they look at the cells on the tissue, and a pathologist interprets the meaning of those and gives a diagnosis. Applying a genetic diagnosis provides a much more detail and understanding of what's going on. One case that I can recall was there was some ambiguity about the the interpretation of the histopathology, and the case was originally thought to be a focal cortical dysplasia brain malformation, but then on genomic reanalysis we identified that it actually had a BRAF mutation, which is associated with a type of low-grade tumour. And so by being able to provide that kind of information, we're able to help guide a change of clinical management and altered monitoring for that individual.
Thanks, Sarah. Another question, Ravi.
Thanks very much, congratulations. I just wanted to know, basically, at the moment, when a child has epilepsy, there's a relatively small repertoire of drugs we can use. How far are we away from actually going to the point of this group of epilepsies will respond to this drug better, and do we need to develop new drugs? And do we have a pipeline to do that?
That's probably a clinician question. Catherine.
Thanks, Ravi. Yeah, so the answer is we can do that for some types of epilepsy, including the most common genetic epilepsy, where we know which treatments are better, but we also know which treatments to avoid, ones that can result in seizure worsening, death, worsened development. So that's really critical for some children. But even then, it's almost like a band-aid. Treating the seizures, not the underlying cause. So yes, we absolutely need better treatments. For many of the other epilepsies, we don't have specific treatments. So I guess we're at different points for different genetic epilepsies. There are a number of different pipelines. There's a lot of development of advanced therapies like gene therapies and ASOs, and we're about to start being involved in some of those trials. There's also options and things we're trying to get set up through our work, and particularly through Sarah's brain tissue work, to look at things like high throughput drug screening and how we might use those to repurpose existing drugs, for example. So watch this space, rapidly changing field.
Thank you.
Well, thanks, team, for that really amazing description of what you're doing.